Hai, Assalamualaikum warahmatullahi wabarakatuh. My name is Nurhanis binti Halil from Bachelor of Science Honours Biology, UiTM Perlis. My project is under the supervision of Madam Sarina binti Muhammad. So, how papaya peel, seed and leaf can be the antioxidant source to human? This part of papaya will review because they contain antioxidant compounds. This figure shows four main bioactive compounds in papaya peels, which are saponins, alkaloids, flavonoids, and phenols. Flavonoids enhance antioxidant activities, normal tissue growth, and renewal and cellular health all around the body. Flavonoid with vitamin C will reduce the oxidative stress. For papaya seeds, there are four main bioactive compounds, which are saponins, flavonoids, tannins, and phenolic compounds. Phenolic compounds in plants can be classified into flavonoid, phenolic acids, and tocopherols. Phenolic acids and flavonoids act as antioxidants and can against oxidative illness. For papaya leaf, there are five bioactive compounds, which are papain, polyphenols, flavonoids, saponins, and tocopherols. Polyphenols compounds have a unique place in environmental sciences as important class of bioactive natural products worldwide. Polyphenol possess biological properties such as anti-cancer, antioxidant, and anti-inflammatory effects. So, why papaya was chosen as antioxidant source in my research? The fruit demands, especially papaya, has increased as the world's population has recently grown rapidly. This will produce high quantities of fruit waste such as peels, seeds, and leaves, may create environmental problems. Besides, due to negative side effects of modern drugs, many people have resorted to natural products instead of modern drugs. BHT and BHA are examples of synthetic antioxidants, but these types of antioxidants have been reported that have some harmful effects. So, why this research is important? Papaya is popular fruit in Malaysia, so there is high consumption of papaya, making its peel, seed, and leaf are generated in large quantities. So, this may create environmental problem. Besides, this research work is to turn this fruit waste into useful product as to obey the law to reduce the usage of landfills by government as it reach their capacity. There are many methods have been established to explore antioxidant properties of a substance. There are two primary approaches. First, direct determination of the substance of individual high molecular and low molecular, and second, based on appraisal of integral substance. The second approach should be considered ideal and more enlightening, and the main most popular integral methods are DPPH and FREP-ACIS. Based on the study conducted by Ng on bar graph in yellow, acetone solvent shows the higher percentage. The extract of acetone showed a great ability to scavenge radicals, a strong ability to reduce, and a potential source of natural antioxidants. The blue color is result from study of Asghar in 2016, shows that methanol solvent has the highest scavenging activity. These data are agreeing with results reported by a researcher who studied three varieties of berry, which show that methanol was the most efficient solvent for extraction antioxidant compounds. The study conducted by Lydia in green color showed that between ethanol and acetone, acetone shows the higher DPPH scavenging activity, different with the study conducted by Marina and Noriham in grey colored bar graph, which compared the antioxidant activity by different concentration of sample. 250 microgram per ml of sample concentration has high percentage of DPPH assay. In the study conducted by Ng, the highest percentage antioxidant activities are in deionized water extract and the lowest is hexane solvents. Deionized water was chosen as the good solvent for the extraction of papaya seeds in this study and was also cheaper in price and abundantly available. Next, based on the study in green color which is by Asghar shows that water solvent has the highest antioxidant activity. Polar liquids such as water are typically good solvent for polar substance. Different with a study done by Sophie which compared between two concentrations of sample. The graph shows that concentration at 400 mg per liter is the higher radical scavenging compared to 200 mg per liter sample concentration. This means that scavenging ability of the samples increase when an increase in their concentration. This bar graph shows the antioxidant activity of papaya leaf using DPPH assay with different type of solvent. Based on the study by NISA in green bar graph shows that methanol has the highest percentage of antioxidant activity, similar to the result reported by other study which show that methanol is the most effective in at a large amount of phenolic content from walnut fruits. Ethanol extraction solvent shows the highest percentage of antioxidant activity for papaya leaf in the study of Asghar in purple colored bar graph. This is also similar to the result reported by Ballesteros in 2014, which stated that the ethanol was considered as the most suitable solvent for the extraction of antioxidant compounds from coffee silver skin. The study by Ng in blue graph color shows that ethanol and hexane solvent has the highest Fred value. Green color bar graph is study done by Dada et al. In 2016, which comparing Fred values between peels and seed of papaya, and papaya seed has the highest Fred value. This bar graph shows the antioxidant activity of papaya seed using Fred assay with different type of solvent. In orange bar graph, a study done by Ng shows that deionized water has maximum reducing power in papaya seeds. Different with the study conducted by Sophie in green color, which compared the absorbent with different sample concentration which are 200 and 400 microgram per liter and using ethanol. The bar graph show sample concentration of 400 microgram per liter is the higher absorbent value, which means its high reducing power. The study conducted by NISA shows that deionized water has higher Fred value than ethanol and methanol. This is also similar to the result reported by Barchan et al. 2014, which showed that water was found to be the most efficient solvent for the extraction to have antioxidant compounds in Mentha species. To conclude, antioxidants mainly from food and medicinally plants have been investigated intensively. DPPH assay is ability of the different antioxidant to donate an electron or hydrogen. FREP assay is to compare antioxidant based on the ability to reduce ferric ion to ferrous ion. The main problem with the current assay use is that usage of spectrophotometer, which is expensive to purchase and it takes time to use. My study suggests that develop more convenient DPPH assay by using a smooth surface, for instance, TLC or paper, a scanner, and the free downloadable color measurement software ImageJ. The ImageJ software will evaluate the spot image to determine antioxidant activities. Advantage of this new method are inexpensive, low consumption of chemicals, and the most important is reduction of the total test time. I'm at the end of my presentation. These are references that I'm using in this slide presentation.